Welcome to the Deep Dive. As you start your journey into medicine, well, you're going to encounter just countless pathways and processes. It's pretty complex stuff.
It really is.
Today, we're aiming to give you a really crucial piece for that biochemical toolbox you're building. We're doing a deep dive into some fascinating energy pathways beyond just glucose.
And importantly, what happens when things go wrong with them?
Exactly.
Yeah.
Our mission here is to take this complex metabolic info, make it stick, use some memorable anchors, metaphors, insights that you'll actually use, all based on solid text and research.
Let's jump in.
Okay, let's unpack this. Glycolysis, it's foundational, right? The main highway for energy.
Absolutely.
But it's not just glucose marching straight to pyruvate, is it?
Yeah.
There are these side characters, intermediates, that do more than just pass through. They seem to have pretty significant roles themselves.
That's a great way to put it. What's really fascinating is how they work as these sort of biochemical junction boxes. They link glycolysis to other really vital processes. Super efficient.
Okay, like what?
Well, take DHAP, dihydroxyacetone phosphate. It's not just a quick stop in glycolysis.
Yeah.
It's absolutely essential for making triglycerides, fat storage in adipose tissue in the liver, and phospholipids too.
The building blocks of cell membranes.
Exactly. It gets converted to glycerol-3-phosphate first, but yeah, fundamental stuff. Think about that cellular intelligence diverting this molecule from energy burning to building structures or storing energy long term. It's quite remarkable.
Wow. Okay, what else?
Then there's 1003-bisphosphoglycerate, or 1003-BPG. Now, this one is really interesting, especially in red blood cells. You find loads of it there.
Why red blood cells specifically?
Because it acts like hemoglobin's oxygen regulator.
Yeah.
Or maybe a mood ring for oxygen affinity.
Right.
It binds to hemoglobin, makes it a bit tenser structurally, and that encourages it to release oxygen to the tissues that need it most.
So it fine-tunes oxygen delivery.
Precisely. It gets quickly converted to 2-XT3-BPG by an enzyme. Bisphosphoglycerate mutates, and that 2-X3-BPG is the key player here. It allows red blood cells, which, remember, have no mitochondria,
Right.
to directly adjust oxygen release based on what the tissues need, right there in the bloodstream. It's like an immediate feedback loop. Very clever.
That is clever. Okay, so if these pathways are so critical, how does the body control them? It can't just be chaos, right? Is it just a simple on or off?
Good question. And no, it's definitely not simple. Glycolysis is finely tuned. Think of it like a metabolic thermostat.
Okay.
The main control points are those steps that are essentially irreversible, the big commitment steps. Step 1 with hexokinase, step 3 with phosphofructokinase, that's a huge one, and step 10 with pyruvate kinase.
So those are the main switches.
Kind of, but there are several dials used to control them. Things like how much substrate is available, product inhibition, where the product slows down its own making.
Like hexokinase being inhibited by its product, G6P.
Exactly. Perfect example. Then there's feedback inhibition, other molecules binding elsewhere on the enzyme that's allostery, and even covalent modification, like sticking a phosphate group on or taking one off.
Right, phosphorylation.
Yeah. So phosphofructokinase, that key enzyme, it's inhibited if there's lots of ATP around compared to AMP, especially in muscle. That signals high energy.
Makes sense.
But in the liver, insulin actually activates it by taking a phosphate off, dephosphorylation. And pyruvate kinase, way down at step 10, is also activated by insulin in the liver the same way.
So here's a thought for you listening. Imagine glycolysis is like a busy factory assembly line. What happens if the raw materials suddenly stop coming in? Or maybe too much product piles up at the end? How would you, as the factory manager, regulate that line to prevent total chaos? Think about those control points.
That's a great analogy. It really highlights the need for regulation at multiple points.
But there's also that cofactor, NAD plus VSOP. It needs constant recycling for glycolysis to even keep going. What happens when the usual recycling plant oxidative phosphorylation isn't available?
Yes. NAD plus silate. Crucial. Normally, it gets regenerated later on in the mitochondria during oxidative phosphorylation that requires oxygen. But some tissues have challenges. Red blood cells, as we said, no mitochondria, no oxidative phosphorylation possible.
Right.
Other spots like the kidney medulla or the lens and cornea of the eye have very few mitochondria. And then there's situations like really vigorous exercise or any tissue starved of oxygen.
Not enough oxygen for the normal recycling.
Exactly. Not enough for efficient oxidative phosphorylation. So in all those cases, anaerobic glycolysis steps in. Lactate dehydrogenase, LDH, becomes the star.
Ow.
It regenerates NAD+ by converting pyruvate, the end product of glycolysis, into lactate. This recycling crew is absolutely essential for these cells to keep making any ATP at all, even if it's way less efficient than the aerobic route.
So it's a survival mechanism.
Absolutely.
Yeah.
Without LDH in those situations, glycolysis would just grind to a halt, no energy, cell death.
Yeah. Incredible adaptation. Yeah. Okay, let's switch gears a bit. Beyond glucose, our bodies can use other sugars too, right? Like fructose and galactose. They seem to have their own pathways, like side entrances to the main glycolysis highway.
Indeed. Very flexible systems. These side entrances are fascinating. Let's look at fructose first.
Okay.
Fructose catabolism happens mostly in the liver. It gets phosphorylated first by an enzyme called fructokinase, making fructose 1-phosphate. Then another enzyme, fructose 1-phosphate aldolase, splits that into two pieces, glyceraldehyde and DHAP. And both of those can jump right into glycolysis.
So a specific route for fructose. But what if one of those side entrance enzymes is missing? What does that mean clinically?
Right, that's where we see problems.
Yeah.
Disorders. There's essential fructosuria, that's a fructokinase deficiency. Fructose just builds up and spills into the urine.
Is it serious?
Usually not. It's rare, maybe 1 in 130,000 births, and generally harmless. Think of it as a harmless overflow, no real symptoms.
Yeah.
But much more significant is hereditary fructose intolerance. That's more common, about 1 in 20,000. Here, the deficiency is in the next enzyme, fructose 1-phosphate aldolase.
And what happens then?
Well, now fructose 1-phosphate builds up in the liver, and that is toxic.
A toxic backup.
Exactly. It causes liver damage, hypoglycemia, and crucially, it sequesters inorganic phosphate. It traps it.
Ah, so the phosphate isn't available for making ATP.
Precisely. That's a huge part of the toxicity. For babies, symptoms are things like poor feeding, vomiting, nausea, abdominal pain after they ingest fructose or sucrose. Treatment. Strict dietary elimination. No sucrose, no fructose.
Got it. Okay, what about galactose? For milk, sugar, lactose. You said earlier it's not just fuel.
Correct. Galactose is vital, yes, for energy, but also for making important molecules, like lactose itself in mammary glands during lactation.
Makes sense.
And also glycolipids, glycoproteins. These are critical for cell recognition, cell signaling, structure, really fundamental stuff.
So how is it metabolized? Also in the liver?
Primarily, yes. First, galactose gets phosphorylated by galactokinase, or GALK, making galactose 1-phosphate. Then, the key enzyme, galactose 1-phosphate uridyl transferase GALT, does this neat exchange. It swaps the galactose 1-phosphate for a glucose unit on a carrier molecule, UDP. This produces glucose 1-phosphate, which can become glucose 6-phosphate for glycolysis, and UDP galactose. There's another enzyme, an epimerase, GNO, that can flip UDP galactose back and forth with UDP glucose, which is important if the body needs to make galactose from glucose, say, for making milk.
Oh, that's quite a conversion process. So if those enzymes are deficient, that sounds like it could be trouble, a real sweet poison scenario.
Precisely. That leads to galactosaemia, a group of disorders. Classic galactosaemia is the most common and the most severe. About 1 in 60,000 births, that's a GALT deficiency.
The key exchange enzyme.
Right. So galactose 1-phosphate builds up, but also another molecule, galactitol. This causes severe damage, liver, brain, eyes. Cataracts are very common. Symptoms in babies are pretty immediate after milk feeding starts. Poor feeding, vomiting, diarrhea, jaundice.
Are there other types?
Yes. Non-classic galactosemia is due to a GLT deficiency. Less severe, primarily causes cataracts because galactose and galactitol accumulate. And there's a very rare galeal deficiency, too.
Now, earlier I posed this question. Why might a baby with galactosemia seem perfectly healthy at birth, but then get really sick after their first few milk feeds?
And the answer is right there in the pathway. At birth, they haven't had any lactose yet. As soon as they drink breast milk or formula, the lactose is broken down to glucose and galactose. If they have classic galactosemia, that deficient GALT enzyme can't handle the incoming galactose.
So it piles up immediately.
Exactly. Galactose 1-phosphate and galactitol build up rapidly, and the damage mechanism is, well, it's nasty. Galactose can be converted to galactitol by an enzyme called aldose reductase, but that reaction uses up NADPH plus H plus A, so you're depleting a crucial antioxidant defense system.
Double trouble.
Right, and this galactitol, our sticky saboteur, accumulates, especially in the eye lens, causing cataracts. Plus, the galactose 1-phosphate buildup also sequesters inorganic phosphate, just like in fructose intolerance, crippling ATP synthesis. It's a multi-pronged attack on the cells.
That makes sense now. Okay, let's shift gears one more time to a pathway that You said doesn't directly make APP, but is still indispensable. The pentose phosphate pathway, or PPP.
Also called the hexose monophosphate shunt, yes.
What makes it so vital, especially, say, for a cell that's dividing rapidly or under a lot of stress?
Well, the PPP is a cytoplasmic pathway, runs alongside glycolysis, and it has three really vital missions. I like to think of it as the cell's utility belt.
Okay, utility belt, I like that. What tools are on it?
It produces three key things. You can remember them with the acronym RNS.
RNS.
R is for reducing power, specifically in the form of NADPH plus H plus R. And it's really important to distinguish this from NADH. NADH is mostly for making ATP. NADPH is for building things and for antioxidant defense.
Got it. N.
N is for nucleotide precursors, specifically C5 sugars like ribose 5-phosphate, essential building blocks for DNA and RNA, crucial for dividing cells.
Makes sense.
And S.
S is for sugars that can feed back into glycolysis, things like fructose 6-phosphate and glyceraldehyde 3-phosphate. So it allows this flexible interplay between making building blocks and making energy.
So even without making ATP directly, it's huge for building molecules and, be stressed, protecting cells from damage. That sounds incredibly important.
It absolutely is. That NADPH plus H plus is particularly critical. Yes, it's used for making fatty acids and cholesterol, but its biggest role, arguably, is maintaining glutathione in its reduced state GSH.
Glutathione, the master antioxidant.
That's the one. GSH is the body's main defense against harmful reactive oxygen species, ROS. These are nasty byproducts of normal metabolism, and they increase a lot during stress. Without enough NADPH to keep glutathione reduced, cells would just get overwhelmed by oxidative damage.
So how does the PPP actually make that NADPH? What are the steps?
It happens in two phases. Phase 1 is the oxidative phase, and it's irreversible. This is where glucose 6-phosphate gets converted to a C5 sugar, ribulose 5-phosphate, and CO2 is released. And crucially, this phase generates two molecules of NADPH plus H plus regiber.
Okay.
The key enzyme here, the main control point, the rate-limiting step, is glucose 6-phosphate dehydrogenase, or G6-PDH.
G6-PDH. Got it.
And its activity is tightly controlled by how much NADP plus versus NDPH there is. If there's plenty of NADPH already, the pathway slows down.
Smart regulation. What's phase 2?
Phase 2 is non-oxidative and reversible. It basically takes those C5 sugars and reshuffles them. It can make ribose 5-phosphate if the cell needs nucleotides, or it can convert them back into intermediates like fructose 6-phosphate to feed back into glycolysis. Very versatile.
Okay. Now, the clinical connection. What happens when that key enzyme, G6-PDH, is deficient? What's the impact, especially thinking about those red blood cells again?
Right. G6PD deficiency is a genetic condition. The enzyme's activity is reduced, so you don't make enough NADPH plus H plus H. And yes, red blood cells get hit the hardest. Why? Because the PPP is their only way to make NADPH plus H plus H.
Ah, they have no backup system.
None at all. They're like these ships carrying precious oxygen cargo in stormy seas. They're constantly exposed to high oxygen levels, generating ROS, and they desperately need that antioxidant protection from NADPH and glutathione.
So without enough NADPH, what's the cascade? What actually goes wrong inside the red blood cell?
Okay, so no NADPH means glutathione can't be kept in its reduced protective GSH form.
Right.
The cell becomes super vulnerable to oxidative damage from ROS. Hemoglobin itself gets oxidized, other proteins too. They clump together, forming these insoluble aggregates called Heinz bodies.
Heinz bodies, like rusty anchors.
That's a good visual. These Heinz bodies stick to the red cell membrane. That damage, plus direct oxidative hits to the membrane lipids, makes the cell rigid, less flexible. It can't squeeze through capillaries anymore.
And gets destroyed.
Exactly. It gets targeted for destruction, usually in the spleen. That premature destruction is called hemolysis.
Now, here's the really interesting part for clinical practice. Many people with G6PD deficiency feel completely fine most of the time. They're asymptomatic until something specific triggers a hemolytic crisis. What are those triggers? What do future clinicians absolutely need to know?
That's the crucial point. These triggers all cause a sudden spike in oxidative stress that the already weakened system just can't cope with. A useful mnemonic is the 3Fs.
Well, 3S, okay.
Fava beans. Eating them can cause a severe reaction called favism in susceptible individual.
Fava beans?
Second F, certain pharmaceuticals. The classic examples are anti-malarial drugs like Primakine and also some sulfonamide antibiotics.
Important drug classes to be aware of.
Definitely. And the third S is fever. Any infection causes inflammation and increases oxidative stress systemically. So an infection can trigger a crisis.
And the symptoms are those of hemolysis.
Yes, signs of hemolytic anemia, fatigue, being pale, shortness of breath, maybe jaundice because of all the bilirubin released from the broken down hemoglobin.
So let's circle back to that question for you listening. Why do individuals with G6PD deficiency often feel perfectly fine until they encounter one of these three Fs?
Well, it comes down to reserve capacity, doesn't it? Under normal baseline conditions, even with reduced G6-PDH activity, they usually make just enough NADPH to keep enough glutathione reduced to handle everyday oxidative stress.
They're coping, just about.
Right, but then they eat fava beans or take primaquine or get an infection. Suddenly, there's this massive wave of oxidative stress. Their already limited NADPH production gets completely overwhelmed. Glutathione levels plummet.
And the red cells are left defenseless.
Utterly defenseless. The damage happens rapidly, leading to that acute hemolytic crisis. It's like a tipping point. They exist near the edge, and the trigger pushes them over.
Wow, what a deep dive indeed. These pathways, they're so intricate, and the clinical conditions when they falter are really profound.
Absolutely. And if you connect it to the bigger picture, understanding these metabolic sort of detours like fructose and galactose metabolism and these utility belts like the PPP, it's just key to grasping biochemical balance.
Yeah.
And it really drives home how seemingly subtle enzyme deficiencies can have massive clinical impacts, especially as we saw under stress or with specific environmental triggers.
We've really seen how molecules like DHAP and 1,4,3-BPG wear multiple hats, linking glycolysis to fats and oxygen transport, and how conditions like galactosaemia, fructose intolerance, and G6PD deficiency just highlight this incredible interconnectedness and sometimes fragility of our biochemistry.
Well said.
So as you continue your medical journey, here's something to ponder. How many other everyday things, foods, common medications, might be subtly interacting with our metabolic pathways in ways we're maybe only just starting to figure out? And how could that knowledge potentially unlock new ways to treat or even prevent disease down the road? Something to think about.